Well, we have the pleasure of having Professor Mark Febbraio on the podcast today. Mark, thanks so much for taking the time.
My pleasure.
Your knowledge on exercise and Myokines and everything you've studied is super interesting. And I was going through the research and realised that I don't have the same knowledge at all, but we do share a passion for sport. And so I won't mention the upcoming Ashes series at all. But how did
No.
your maybe at the end. How did your sort of love for sport translate and sort of get you to where you are today, where you're studying, the different signaling molecules
I,
of muscles and its various effects?
so, Ben, I had a really interesting uh, introduction into um, academia, actually. I was not really interested in science at all at school, at all. I was interested in sport. I still am crazy interested in sport. And I was um, I was really in high school, did mainly humanities rather than science subjects. And the only thing I ever wanted to do was to be an athlete. And if I couldn't be an athlete, or even if I could, to write about it. So to be a sports journalist. So in school, I really did all humanities subjects and pretty much excelled at subjects such as English and history, etc. And my first degree that I started was actually a journalism degree. And at that stage, I just started to get in a triathlon, and I thought I can read about and write about sport and do sport. And in journalism, I had a very good friend, Tim Baker, who actually had the same idea, and he went on, and he was a surfer, and he went on to becoming the editor in chief of Tracks magazine, writing books on surfing and basically chasing the dream. I couldn't really get a cadetship, so in order to continue with journalism, you needed a job, and I couldn't really get one. I didn't have many connections, so I went to. I decided I'd do a human movement degree to learn more about the physiology of exercise, with the hope of going back and freelance writing. But once I got into my degree, I fell in love with science. And I remember walking my lecturers to their car, and they used to sort of tell me, "Oh, Jesus, mate, give me a break, will you? I just want to go home." And 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 so I became very interested in science, but I was still much more interested in pursuing my athletic career, which I did after my degree. I took a few years off, and then I was racing on the international circuit, an international triathlon circuit, and I was on a team, and I did it. I had done Hawaii a couple of times, Hawaii Ironman a couple of times, and anyway, I went and I did a race in Japan, in Lake Biwa in 1990, and I became very distressed during that race, very dehydrated, very heat stressed. I was in a bit of strife. Finished the race in really terrible shape, in hospital. And then I decided, I sort of had an epiphany that I wasn't good enough anyway to really make it, which looking back was a bit of a stupid thing to say because I was only 24 years old, but patience wasn't my virtue. And so I went back and did a PhD. I went and saw my PhD advisor and said that I wanted to do a PhD in the effect of environmental temperature on muscle metabolism, because I had really suffered during this race in the heat. And that's how I got interested in it.
So good. I feel there's anyone who's experienced and enjoyed exercise for a longer period of time, I think intuitively knows that it's doing more than one thing to the body, right? It makes you feel good. I don't know. It changes your perspective on how you think about life, right? So it's doing things for the brain. It's doing things for all sorts. For somebody who hasn't heard of a myokine, or is it myokine or myokine?
My kind.
Myokine. Okay. For somebody who hasn't heard of a myokine, what would be the sort of entry level understanding of it? Like, what isn't myokines?
Okay, a myokine is very simply a, a a molecule, a substance, a protein, or a metabolite that is released from the muscle during contraction that has an effect on another part of the body by engaging a receptor or by activating another molecule by getting into another cell. So that's basically a myokine. And we came up with the term myokine because my colleague in Copenhagen, Bente Peterson, and I were studying the effects of exercise on a cytokine called interleukin 6. And we were studying this independently because many groups had shown that interleukin 6 went up during exercise in the blood. But everybody just assumed that it was that its cellular origin was the immune cells. And that was a fairly, not an outlandish assumption because immune cells are the main cells that make this molecule. But I had a very talented PhD student around the turn of the millennium, Rebecca Starkey. She's now a medical doctor. And she came to me and said, nobody's ever really shown that it's the immune cells that in fact make interleukin 6 during exercise. It's just assumed. So we went and did a series of studies and it turned out that it wasn't the immune cells at all. And so by elimination, we had come to the conclusion that it was the muscles that were making interleukin 6. And Bente Peterson independently had data on this and presented the data at a conference. I was a very, very junior PI. PI is a principal investigator, lab head. I was very junior and I was a bit worried because I thought, she's a very, very established scientist. And I said to Rebecca, what should we do about this? And she said, let's go and talk to her. So I went and talked to Bente, and Bente was, rather than, saying, you're my competitor, she embraced the idea that we would work together because she was principally a human clinical scientist and I was more a basic scientist. So we've collaborated for the last 25, 30 years and continue to do so. And the myokine field sort of has exploded a little bit.
It seems really, really complex because, like anything in the human body, right, you can't really do one thing. So whether there's like a muscle contraction and you stimulate the release of these myokines, so many different things happen downstream of that, or at least my understanding of trying to grasp your research. So maybe if we start with IL-6, my understanding is it's sort of a bit of a hero and a villain at the same time. So it's sort of
absolutely.
Good. Can you explain that? Because
I can. It's a little difficult to explain, but I'll try and do it. So the reason why interleukin 6 is a bit of a hero and a villain is because it signals in two different and distinct ways. One is called membrane-bound signaling, where it binds its cognate alpha receptor, and then engages two other, what we call beta receptors, and then transduces a signal. And that seems to be a normal physiological process that is a very good thing. For example, if you want to activate platelets, that's how you do that. But it can also be a very pro-inflammatory molecule, and it signals in a way where the interleukin 6 receptor can get cleaved, and that can become soluble. So you have a soluble interleukin 6 receptor. Then interleukin 6 can bind to that soluble receptor in the blood, and it can then, through processes we're not exactly sure of, it can go to cells that express the beta receptors, the SGP130 beta receptors, but not the IL-6 receptor. And they tend to be the very pro-inflammatory bad cells, and you get a process that's called trans-signaling. Now, to give you an example about how this has progressed, I have developed a membrane-bound signaling um, drug that appears to be very positive for many diseases, but there is a drug in phase 3 clinical trials called elamcocept, which blocks trans-signaling. And we've done some experiments where if you put the two drugs together, they work very, very well. So one activates IL-6 in one sense, and the other blocks IL-6 in another sense. And so that's why it's so complex, because of this complex dichotomous signaling process.
Right. Okay. So the one drug sort of upregulates all the good with R6, and one dampens all the bad, and so you're just left with the
Correct.
good at the
Correct. That's correct.
end.
That's in simple terms.
Okay, understood. So what are the downstream use cases of be it through exercise or be it through the combination of drugs that could lead to favorable outcomes for for the various different people with R6?
So one of the things that we found that IL-6 can do is it can act. This is some very early studies. Um, it 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 can do two major positive things that we've published in the past. One is it can actually activate a protein called AMP activated protein kinase, or AMPK. And AMPK is a protein that gets activated during exercise. And what AMPK does, it upregulates fat oxidation. So IL-6 is one of the things that increases lipolysis and fat oxidation during exercise, and we've shown that. The other thing AMPK does, it can increase glucose uptake into a cell. So it can increase insulin sensitivity, for want of a better term. And we showed that. But the other thing that we showed early on is that during exercise, interleukin-6 can act in a way of being a metabolic danger signal. So if your muscle cells are running out of glycogen, or sugar, which fuels the cell during exercise, it sends the signal from the muscle to the liver to liberate more glucose, put more glucose into the blood, and therefore you can use that liver-derived glucose as a substrate when your muscles start getting glycogen deplete. So it acts as a signal, a crosstalk signal from the muscle to the liver. And we were able to figure this out because we did a study where we exercised both legs, but we had the patients perform one-legged exercise the night before to deplete that leg of glycogen. And then when we were able to test this, we showed that the leg that had depleted glycogen released more IL-6 and released it earlier than the leg that had more glycogen in it. So that was sort of proof that it was a danger signal being released from the muscle.
Oh, right. And actually, just a side tangent. Did the leg that depleted more glycogen have some sort of like super compensatory effect when glycogen was put back in to the system?
Well, no. Basically, what happened was that the glucose uptake by that leg went up, and that leg was able to use the glucose from the blood in order to maintain contraction and not fatigue.
Right. Wow. And is the process is that gluconeogenesis when the signal gets converted to the liver?
Well, gluconeogenesis is a process that takes place at rest, and paradoxically, if you give interleukin-6 to people just during rest, it doesn't really upregulate gluconeogenesis. what it does, it accelerates hepatic glycogenolysis during exercise, which is slightly different. So, because if you were to increase gluconeogenesis at rest, you would actually induce metabolic disease because you put more glucose into the blood when it's not needed. And that was one of the things that people said. They said, "Oh well, look, you showed that interleukin 6 increases hepatic glucose production. Well, there you go. It must be bad." But when we did the experiments at rest, that was not the case.
Yeah, it's contextual, right?
Yeah.
What seems to be amazing is this is just one. This is one downstream effect of one. Is interleukin a myokine or a cytokine?
Yeah, it is. It is. It is a myokine because it is released from skeletal muscle, and it does act on another tissue. Yes, it is. And I'll go ahead and ask your question.
Oh, I mean, tangents galore. Please go ahead to go off in any which way. Good.
So once we sort of found this, that's what we wanted to do. We wanted to examine, what else could be coming out of muscle, or not just muscle, but other tissues, adipose tissue, liver. There are adipokines, there are hepatokines, there are batokines, proteins that come from brown adipose tissue. So one of the things that we've been doing the last decade, together with Dr. Martin Whittam from the University of Birmingham, he was actually did a landmark study in our laboratory a few years ago now, and one of the challenges about studying, like trying to do a proteomic screen in blood, is that plasma contains vast amounts of a protein called albumin, and it's very sticky. So it's very difficult to capture all of the proteins in the blood. So one way that we got around this is we started studying these vesicles called extracellular vesicles, or they've been referred to as exosomes. They're tiny lipid-layered particles that are released from all tissues during physiology or pathophysiology, and they contain enormous amounts of cargo, microRNAs, proteins, lipids, lots of things, and they are considered to be key in inter-organ communication. So what we did is we've been studying these EVs in the context of exercise, and we've been able to show that during exercise the EVs go up quite markedly in the blood, and more importantly, what's in them can change during exercise. So the proteomic profile can change, for example. And we think that these are key to the protective effects of exercise in pathophysiology. So we think, for example, one of the things that we have shown is and published is that in a mouse model, and I have to say that all of the studies that we do so far to look at how exercise can prevent disease from a mechanistic point of view are all done in rodent studies, because we can't transfer EVs from human to human yet, but we can transfer them from mouse to mouse. So if we purify EVs from a mouse that has previously undergone exercise, and we transfer them to mouse models of various diseases, then exercise does have a preventative effect. And we've shown this in published work in a mouse model of Alzheimer's disease, and we have unpublished work in a mouse model of fatty liver disease and a mouse model of breast cancer, that they can have protective effects.
Yeah, I did read a study where exercise blood put on a tumor was more favorable than just normal blood put on a tumor.
Yeah, yeah,
I'm
and we've shown that as well. And one of the things we showed in this, we're hoping to publish this soon, but one of the things we did show in this study where we transferred EVs purified from an exercise mouse into a mouse model of breast cancer is that the actual EVs were enriched in proteins that prevent metastatic cancer. So they had an anti-metastatic niche. And now what we're doing is we're exercising newly diagnosed women with breast cancer to see if those proteins are actually enriched in their EVs.
Wow. Okay, let's go to exercise oncology then. We'll come back to inflammation, but then maybe they're one and the same thing. I don't know. Maybe they're linked. Um, that does seem to be the complex thing with what you do. I imagine if you're trying to isolate one myokine, you probably find it's hitched to something else as well, and it's there's so many variables, right? So that must be quite hard to sort of isolate one thing
It's
that's doing it.
it's very challenging. I mean, um, the the epidemiological data in humans is very clear for about seven or eight different cancer sites, but the randomized clinical trial data um, is very scant. So there are very few studies where there's been random clinical trials um, where there's one group of patients that have undergone standard care and another group that has undergone standard care plus exercise. But a paper came out earlier this year. I'm pretty sure it was in colorectal cancer, and they did such a study, and there was a protective effect. The challenge is coming up with mechanisms to then try and develop, um, for want of a better term, exercise mimetics to, to try and see if you can put some of this information in a therapeutic context, because it's and that's why we're trying to see, can a patient store their own EVs and be infused with them at a later date when they can't exercise. Because the difference between, say, exercise and cancer, as distinct from exercise and, say, metabolic disease, is that now if patients are undergoing, multiple bouts of chemotherapy, then fatigue is a factor. And sometimes they just would be unable to perform exercise when they're feeling the effects of chemotherapeutics and maybe being fatigued. So coming up with mimetics or therapeutics is a useful thing to do. But as you said, Ben, the complexity in the system and the complexity in systems biology it's something we really don't understand very well at the moment. We think we do. We always, as scientists, think we know more than we actually do. We still don't know a lot about biology, and we don't know a lot about medical oncology. We know very little actually about the mechanisms by which exercise can protect against disease. And sometimes exercise can't protect against disease. And the other thing is we can't ever think that it's a one size fits all. Our genetic makeup is such that there are some people that would respond very well to a therapeutic or to a diet or to exercise, and you give that diet or that therapeutic or that exercise regime to the next person, and they don't respond at all, because everything is a complex interplay between the environmental stressor or the environmental interaction and the genetic makeup of the organism.
It's such a good point. I mean, it's funny. I always find that I get to speak to brilliant people like yourself who have been studying, a specific niche for a number of years, and the consensus is always once you've had enough time in a field, is actually, you know how much we don't know. I mean, one of my neighbours is actually he's a professor of psychiatry at Oxford, and I love speaking to him because it's usually like, well, it could be this or it could be that, but actually we don't really know. And it's just this, they've taken the time to really understand something. We're actually pretty humble and actually appreciate what we don't know. But
Well,
anyone touting
uh,
certainty probably hasn't done it yet.
it's so true, and if you think about, on one hand, people could argue, oh well, science hasn't been very successful because we haven't come up with blockbuster therapies, but if you think about, I mean, I'm a little bit older than you, but if you think about in your lifetime, there have been at least that come to mind at least two completely transforming therapies. One is checkpoint inhibitors for cancer, and the next one is GLP-1 receptor agonists for treating obesity, which is probably going to win some of my colleagues the Nobel Prize at some stage. Um, and 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 it takes, as the saying goes, it takes a village. It's not just those individuals who who there's those individuals who get the accolades, but it's just a whole field of toil and research to come up with these breakthroughs. I mean, if you think about the discovery of insulin by Banting and Best 120 years ago, just transformingly saved people's lives. If you think about McFarlane Burnett and Fleming, Alexander Fleming, and their discovery of penicillin, we just take all of that for granted now. So,
Mm.
these it only takes a handful of discoveries, despite thousands of tens of thousands of hours spent by scientists trying to come up with things. It's okay to fail, but we are making inroads into all the time into understanding or expanding our knowledge more and more, particularly in the area of tissue crosstalk and systems biology.
Absolutely. I mean, I really enjoyed the anecdote of someone who could have been your competitor, quote unquote, but actually it's just, it's not a zero sum game, right? Knowledge shared is super beneficial, and as a result, a favorable outcome came from it, right? When you talk about
Correct.
exercise, it's such a broad term, which could be, slow zone two. It could be sprint training. People could do a form of Pilates. It could be weight training. You name it. What type of exercise is, if any, is more favourable for maybe, let's say, sort of cancer prevention slash uh, avoiding a cancer recurrence? Do we have that data yet?
Well, I mean, in terms of, I mean, the greatest risk factor for cancer development is age. The greatest risk factor for most diseases is age. So, in aging, you have a decline in VO2 max, in aerobic fitness, and you also have a decline in muscle mass. So, in clinical oncology, what is typically uh, prescribed is a combination of aerobic exercise and resistance exercise. And I myself, um, used to loathe going to a gym and lifting weights, but as a 60-year-old, I have to do that now, and I've had to do it since, because one of the things is that the decline in mass is associated with frailty, and frailty is associated with premature death. One thing I will say in our animal studies is that it seems to be the more exercise you can do, the better. So if we give mice running wheels. Mice love to run. They're nocturnal, they love to run. They run a black mouse, a C57 black 6 mouse, which is a strain of mouse, will voluntarily run between 3 and 20 kilometres a night.
Really? That's
which
absurd.
is astonishing. And
Yeah.
when we implant the tumours and we give the mice either running wheels or running wheels that are locked. So the environment is exactly the same. The only thing is that they have an environment where the mice will jump onto the running wheel and can't run because it's locked. Now, the mice that exercise have a better outcome when it comes to delaying tumour progression and delaying metastases. If you stratify the data where you take the mice that run, say, more than 6 kilometres a night to those that run less, the data are very clear. The more you run, the better the exercise is in terms of preventing tumour progression. So it's really clear.
Right.
I mean, the mice don't know. It's not self-reporting. We know. We monitor the wheel, and a mouse has its wheel. So we know exactly how much a single mouse runs per night. Now, one of the things that always intrigues me is how come they don't run all the same amount, because most of them are genetically identical, and a lot of them are sibling pairs, they're litter mates. So why some would run more than others is beyond me. That's a challenge in itself. But it's very clear from our data that if at least if you're a mouse and you do more exercise, it's better for you than if you do some, but some is better than none. And actually the human studies that have been done with respect to all-cause mortality seem to suggest the same thing, that if you do more exercise, the only cancer actually that is negatively related to exercise, and I often ask people what it is, and they don't know what it is, but when I tell them, it's very clear, and that's melanoma, because exercisers usually spend more time outdoors in the sun.
Oh,
So,
right.
so that's the only cancer where there seems to be a negative effect of doing a lot of exercise. The other
Wow.
cancers are either it's not affected or exercise has a benefit.
So I always find that if there's what the research says, and then there's someone like you who interprets the research and actually, it changes how you, as a exercise fanatic, right? You're passionate about exercise. Actually alters your day-to-day behavior. So knowing what you know, how many strength sessions do you try and do a week, and how much endurance do you try and do a week?
So basically, exercise has to, fitting things into your life has to be, there has to have some context to this. Now, I can't run anymore at all. Christmas Eve 2018, I was on my bike, and I was in a fairly horrendous interaction with a car,
Oh,
and I wound up in pretty busted up. But mainly, I have completely shot knees now. My knees are completely shot to pieces. I have no meniscus left. I have no cartilage left. Yeah, so I
Wow.
can't do. So I can't run. So what I have my wife and I. She's also a professor. She has trouble running because she has like turf toe. So we actually religiously walk every day. So we walk every day, and we have two dogs. So we take the dogs between. We average about 6 kilometres a day every day. So we walk every day. So that's one thing we do. And then I try and cycle and resistance train every other day. So, about three times a week, 3 to 4 times a week on the bike, and 3 gym sessions a week, and walk every day. So average 15,000 steps a day
Wow.
for the last three years. So, you just
Yeah.
try and make, to people out there, don't think of exercise as drudgery and as something that's, something you have to do but is not pleasurable. Pick things that you like to do, because otherwise, ultimately, if it's not nice, you won't do it. It's like eating food that you don't like.
Mm.
You won't actually do that if it's not pleasurable. So my advice would be but also, I mean, one thing I was notorious at doing when I was younger was overtraining, and that can be as bad as doing not enough. So people need to listen to their bodies, and if they're tired, it's okay to have a day off, or it's okay just to go for a walk and not smash yourself every day. But
Mm.
this is not based on science. This is just based on N of one. Um, my
Yeah.
my thoughts on being a lifelong exerciser. It's not based on anything scientific.
Sure, sure. I mean, I think often the scientific stuff informs, how people actually sort of try and go about it. Life gets in the way and things like that, right? But as you said, you know, within the context of a mouse or cancer, it seems that more exercise is better. So then, if someone wants to be quote unquote as best as they can, it seems like riding that sweet spot as much as you can do without dipping into overtraining. Almost like this
Yes.
J curve I've heard you speak about
Oh
is
yeah.
the sort of
There is absolutely, Ben. There is and that's the hardest thing for athletes to do. The hardest thing for athletes, I think it's harder for athletes, at least the athletes that I was associated with, and I was a notoriously hard trainer because I was talentless. So I never had a talent that some of these other triathletes had. I didn't have the genetics, so I I used to have to train really hard, and then the hardest thing was when do you when do you when do you recognize that to do this next session is actually going to be detrimental. Now, this was eons ago when I was an athlete. The tools that coaches and sports science has now is so much more developed. Like there are blood signatures being developed for Tour de France riders. I've seen data on this um, where each individual um, can have their um, blood proteome screened, and there could be um, an enriched biomarker signature that's telling the coach this athlete is starting to overreach, or this athlete is starting to be going into a phase of hyponutrition. So telling the coach this athlete needs to increase their caloric intake a little bit, or has to back off their training a little bit. I mean, these
mm,
things are being developed in sports science. This is not my field, that I just see. I find, if I go to a conference and there's a really applied sports scientist speaking, I'm very interested in it, not for my science, for my, prior life and my general interest.
Yeah, 100%. I mean, me too. I used to play rugby in Australia, and sort of like an ex-girlfriend never quite got over. Right, I'm always quite interested to see what's going on in the world, and yeah,
that
especially
that's a that's a really good way of putting it. I've never got over not being an athlete.
oh,
I'm very satisfied with my scientific career, but I've never got over not being an athlete.
oh
And this
God, I
is,
feel
and this is 35 years of pain.
oh God. Well, I can relate in my own way, perhaps. What I'm confused about is inflammation. So the whole piece of they used to do, or athletes do a lot of recovery in ice baths after exercise to help them recover. But if you're going for like you're trying to put on muscle, that inflammatory response off the back of weight training is favorable. So you don't want to get in the ice bath after. So it confused me around general inflammation and exercise because my understanding was exercise is great for inflammation. Most chronic diseases have an inflammatory aspect to them, and exercise seemingly lowers inflammation, but yet in some contexts raises inflammation. How does exercise and inflammation actually? How does that happen, and what is the actual truth to it?
I think you've just given the best answer that I could actually give. Exercise can be anti-inflammatory. We did this very, very cool study over 20 years ago. We could only have done this in Copenhagen with my colleagues. So a very, very unequivocally pro-inflammatory cytokine is TNF. And TNF actually doesn't really go up with exercise unless this is where you see TNF going up with exercise is when marathon runners get a syndrome called leaky gut. So they get microcontusions to their intestinal epithelium and they get systemic endotoxemia. Now, this also happens if people have a really, really poor diet with lots of cholesterol and lots of fructose. They'll get systemic endotoxemia. And of course, endotoxemia is LPS, and LPS binds to a receptor on an immune cell called TLR4, and the end product of that is TNF. So what we did in this study is we gave patients, volunteers, a small dose of lipopolysaccharide of bacteria by injection, and we monitored TNF levels over the next 4 hours, and they went up. And then what we did is we did the same thing, but we exercised the people. And when we did that, TNF did not go up. So exercise is clearly, from these studies, acutely anti-inflammatory.
Can I ask
But
what
you're
the
right.
type of exercise was? Sorry to cut
Yes,
you
it
off.
was. It was 2 hours. 2 hours of bicycle exercise in healthy
Okay,
young volunteers, right? But if you exercise too much, particularly as I said in Ironman Triathlon or Marathon, you can actually get systemic endotoxemia. You can get it's again yin and yang, too much of a good thing. Now, when it comes to the inflammation in muscle after a bout of resistance exercise, then yes, that is necessary. Inflammation is necessary. It's necessary to remodel a damaged tissue. So if you, here's an example that there was a very um, important study that was performed by I think Michael Risto was the last author. What he did was a very, very classic study. Exercise produces reactive oxygen species. And reactive oxygen species and reactive nitrogen species is what leads to DNA damage. So people are always taking antioxidants. That this study showed that this was probably not a very good thing, because what they did in this study is they put a group of individuals on an exercise training regime. I think it was, I can't remember, it was a long time ago, but I think it was 8 to 12 weeks of endurance exercise training, with and without the ingestion of antioxidants. And so there was a washout period. It was a double-blind crossover design study. When the subjects were given the antioxidants, they had a blunted training response to exercise. So it was actually a bad thing.
Mm.
And when they received the placebo, they had a much better response. So I think your opening statement about this topic is the answer. We really, it's very complex, because you don't want too much information, but you don't want to block the physiological response to exercise. So physiological ROS production is actually necessary. But if you had a pathophysiological ROS production, that'd be not such a good thing. So it's not a very easy concept to navigate around, because inflammation is necessary. But if you have unchecked inflammation, for example, in autoimmune disease, the biologics to treat autoimmune disease all block out of control inflammatory cytokine storm.
It's complicated. It goes back to the whole thing. I mean, there's this. I was speaking to someone the other day who referenced epilepsy, and how we know the elevation of ketones in the blood in people who suffer with epilepsy, it does do wonders to decrease the rate of seizures. Do we know how it works? Absolutely not. No idea, really. And it feels like it feels like a lot of exercise where people, well, like me, I'm clawing at like, okay, I can get this benefit, I can get this benefit, I can get this benefit. But it seems like there's probably thousands we have no idea about, and lots we've sort of got a rough idea about, and then a few we can sort of isolate to, like inflammation, where it sort of depends. So again, just it's probably more complex than we think.
Yeah, it's very complex. I mean, getting back to your point about what sort of exercise, etc. Benter and I have just written this review. It's actually not published yet. It's still in peer review, and we were sort of trying to think about, well, how can we get more information about how people increase not lifespan but their health span. How do they have healthy living, etc. So I went and researched. I went and did a lot of reading about blue zones. Blue zones are zones where there seems to be a cluster of people that live long, healthy lives. And and there are blue zones in Japan, um, in Sardinia was the first one that was located. And the thing that always seems to there are a few things that link these blue zones, and one of the things is sort of optimal climate. So they're outdoors a lot, but but a degree of isolation. So people tend to have to walk a lot, and these blue zones always seem to be around hilly terrains. So walking up and down hills, gardening, and eating, and the diet seems to be key to this as well. There's no fast food, etc., etc. So I think it's not just exercise, but it's it's so many things that contribute to overall health.
Mm.
One of them being exercise.
Yeah. Such a salient point. The on the whole sort of chronic disease piece, I'd love to understand more about BDNF, and how that gets. Is BDNF a? Is it a myokine? And then what is the link between BDNF and Alzheimer's prevention, if any?
Right. So BDNF is strictly not a myokine, because in the studies that we and others have done, yes, it's produced. Some studies, including ours, suggest that at least in animals, that it's produced by that pro BDNF is produced by muscle cells. Some other groups recently haven't found that, so that's controversial. It does go up during exercise, but it doesn't come even in the studies we did. The origin of BDNF during exercise is from platelets. Now,
Mm.
not our group, but other groups have shown that it can be taken up by um, by the brain during exercise, and it's a neurotrophic factor. So it's very much linked to to uh, cognition, and it seems to go up in the brain during exercise, but it's not a muscle to brain axis.
Okay.
People think it is, but looking at all of the literature, it doesn't. There's not really a suggestion that it is, but it certainly goes up with exercise, and it seems to go up from other tissues. So you can call it an exokine, but not a myokine, I don't think.
Okay, and that's the distinction, I guess. So if it is downstream from the muscles, it's a myokine, but if it's a downstream from just
Yeah, if it
exercise in
comes
general.
from muscle, yeah, it's a myokine. But there are several proteins that go up during exercise that come from the liver, from adipose tissue, um, from uh, blood mononuclear cells, from platelets, etc. I mean, they're still exercise-induced proteins. They're just not coming from muscle.
Mm. Do you think the hype around exercise and brain health in general is overhyped or underhyped at the moment?
I think it's hype just enough. I mean, there is enough evidence to suggest that exercise is beneficial for the prevention of cognitive disease. I think we have so many epidemiological studies on this, and in mice, we have a lot of interventional studies. I mean, exercise induces neurogenesis, the formation of new neuronal cells. It can prevent, glial cell formation in the brain, which is, pro-inflammatory. There is, and one of the hypotheses around um, cognitive disease. It is not only the accumulation of beta amyloid and phosphorylated tau, which are thought to be the primary causes of dementia, but a change in the or a maintenance of mitochondrial integrity in neurons. Now, exercise seems to have a positive effect on that. I think um, I think the things that you need to do to prevent dementia, number one, you can't do anything about genes, but pick your parents. Um, there's a link to obesity and poor diet. So don't be obese. Don't have a poor diet. Don't sleep too little or too much. Uh, and exercise your brain and your body. They're the things that, in terms of, because we are an aging society, and dementia is going to be it already is a paper came out last week saying that it's the biggest killer of Australians now.
Is it? Wow.
In 2023, it was the number one cause of mortality, which sort of makes sense because we're getting better at treating and preventing other diseases,
Mm.
So, but yeah, they're the many things that I think we should all do. And I mean, you don't have to be Einstein to work that out. Do crossword puzzles or quizzes, stay lean, have a good diet, don't sleep too little or too much, don't drink too much alcohol, so diet related, um, and exercise every day. And then
Mm.
um, my mother is 89. She'll turn 90 in about 8 months, and thankfully, she is amazing when it comes to her physical and mental health. And she actually goes to the gym as an 89-year-old Monday to Friday and does water aerobics every day. So, whether it's her genes and she would be cognitively with it in the absence of doing that, I have no idea. But she is never been obese, never had metabolic disease, worked in retail till she was 70 and has exercised every day. So, I mean, she's for me, and again, N of one and hand waving, but she for me is the role model that I'll try and continue to be like.
That's great. That's really cool. What the you've chosen, you've picked, done your own prescription if you've chosen the right parents, right? The
Well,
uh,
one of them, the other one died when I was 20 years old of a myocardial infarct,
oh
and
God, as soon as I said,
and
I
I'm
shouldn't.
and I see a cardiologist every 6 months. So um, cardiovascular disease can can largely be preventable if you take a statin, if you have blood pressure, you take blood pressure meds, if you exercise, if you stay lean, if you don't smoke. My father
Mm.
was a smoker, and statins weren't invented when he died, so
Oh,
um,
right.
it's a little different. But um, but certainly I have familial hypercholesterolemia, and that's something that can only be treated with a statin if you actually respond to statins.
Okay, understood. Yeah, my mum this time last year got diagnosed with stage 4 colon cancer and got the all clear in
Ah.
February, which was,
Oh,
you know,
wonderful.
I know it's good, but now, she plays a load of tennis, which is great. She's healthier than she's ever been at the moment. And I'm nudging her to, pick up the weights and do all this. So after hearing this,
Yeah.
various different benefits of exercise, I'm sure it might be a good nudge. Mark, I speak to you all day. You're a super fascinating man. Um, but appreciate, vastly different time zones, and you've got better things to do with your evening than talk to me. But love to end with some quick fire if that's all good.
Yeah, it's fine.
Love it. So if you could run any study in the world, right, and there's no budget constraints, there's no participation constraints, everything, there's no ethical constraints, there's all good, you can do what you want. What study would you do and why?
Okay, so this is recency bias, but I would do this study. One of the things that we don't know um, very well is why do people appear to lose a vast amount of muscle mass when they go on GLP-1 receptor agonists such as Ozempic? We don't know. We think exercise can prevent this, but we don't know. So the study that I would do is to take a group of patients that are obese, put them on Ozempic, monitor their decrease in food intake, monitor their loss of fat mass and loss of lean mass, and take muscle biopsies and see what exactly is happening to the protein synthesis pathways and the protein degradation pathways in these people. Then I would do a let the drug wash out, let the people regain the weight, and then give them an isocaloric diet that they were on when they were on the Ozempic to see if it's diet alone or something sinister with the drug. Because they're wonderful drugs, but I fear, and we have millions of people on these drugs, but I fear that these drugs could be having a really unwanted side effects in muscle that could lead to age-related sarcopenia. So sorry for the long answer, but that's the study I
Absolutely
would
not.
do.
No, it was fascinating. I'm really interested in the GLP-1 agonists as well, and I the when mum got cancer, I dove into all the sort of cancer cachexia research, and
Yeah.
dove into all the different things that can keep muscle on a human when they're going through chemo and things like that. And I would love to understand if a lot of those things, like HMB, like creatine, maybe some SARMs as well, could be beneficial
Mm-hmm.
in actually preventing that muscle loss whilst on a GLP-1 agonist. Because it just seems like that's the
Well,
last sort of side of them, doesn't it?
yes. Well, cancer cachexia and anti-cachectic therapy is a massive interest area in clinical oncology, and so far we've come up with really nothing.
Really? Well, why
Mm.
do you think that is?
Because we don't understand enough about the etiology of the cachexia, I think. I mean, the thing is, um, you can give some um, some drugs that like myostatin inhibitors or activin inhibitors, and they could work. The problem is that they could also cause enlarged heart, and that's probably not a good thing when you're trying to fight a disease.
Mm. My favorite thing with myostatin is if you do Google, maybe for the audience, you've probably done this already, but for the audience who aren't familiar with them, if you Google myostatin frog or myostatin dog, you'll see what we're talking about. They're just the sort of the most jacked
Or
out.
the best one is to look at the Belgian blue cattle.
Oh yeah,
They have a mutation, and go and have a look at this bull. It is amazing. The
really?
musculature on this bull. Yeah.
Yeah,
A
can
Belgian blue
confirm.
cattle. Go.
That's
Yeah,
ridiculous.
have a look at that.
Wow, wow. Okay, well, yeah. I mean, definitely. This I can see why you bring that up. Fascinating point. When it comes to I guess being a scientist and being in the weeds, often it's you're presented with things that are quite contrary to the consensus. Um, do you believe anything, or do you have anything that you think is true that your field would actually think it would disagree with, but you're pretty convinced it's right?
Um, the answer to that question, I'll answer that question with something that I often say. I don't believe anything. Um, belief, and I don't want to offend anybody who's religious here, but a belief is a faith. Um, all we have in science is to accept the evidence that we have on balance, and that could change tomorrow. So the biggest roadblock that we have in science is scientific dogma. Science changes from day to day. You know, you could. I used to be of the mindset that blocking inflammation could have therapeutic utility in metabolic disease, till a myriad of studies came out showing it does nothing. So I really don't like the word believe in science. I'll answer it that way. I have no
That's,
beliefs. I have
that's a great answer.
opinions based on I have opinion based on what's currently known in the scientific literature. I have
Mm.
opinions, not beliefs.
Well, that probably makes you good at your job, right? You know, strong opinions or strong opinions loosely held, right? Um,
Correct.
okay. Well, Mark, thank you so much for doing this. Honestly, I learned a bunch, and yeah, super insightful. I always know it's a good episode when I want to go and put into practice immediately the topic of discussion after the podcast. So you know, it's not even 9 o'clock here. I'm actually going to go on a run straight away after this, just to
Oh,
you
good
know.
on you, Van. I wish I could run, but as I told you before, I'm limited to walking now. But I may get my knees replaced, and then we'll see.
Okay, great. Well, my dad's got two big titanium knees now after you know
Yeah,
multiple different accidents and various. So he swears by it. But Mark,
yeah.
where can we keep up with your work? Where can we follow along and pull deeper on this thread?
I think basically, I mean, I don't do a lot of lay pieces. It's really I'm flat out being a scientist, but good old PubMed would be the way you could follow.
Love it, love it. Well, we'll put links
All
to
right,
everything we discussed in the show notes, and yeah, just thanks so much for doing this. Really appreciate it.
no worries. I enjoyed it.